Okay, very good morning, everybody. I'll just,
Good morning, sir.
yeah, good morning. I'll share my screen
Yes, sir.
for today's presentation. Morning. Yeah, just starting. Can anybody tell me where we stopped yesterday so that I can continue?
Moving boundary electrophoresis.
Okay.
Moving and zone electrophoresis.
Yeah, I think I showed you one slide where I had written,
Boundary electro.
hello. Yeah, two different important techniques in electrophoresis. One is the moving boundary electrophoresis and the other is zone electrophoresis.
Zone.
Okay. And then I tried to describe something about the moving boundary electrophoresis. I told you that there is no supporting medium as such. Okay. The sample components are in a buffer. Okay. We have a sample mixture and we apply the current directly inside the buffer. Okay. So separation of the sample components by virtue of their electrophoretic mobilities will take place within the buffer. Okay. As the components move, the components which are having similar properties means what? Similar size and similar charges. Okay. That is the similar charge distribution. These molecules will form a boundary. Okay. And depending upon the number of different entities which are present or different species which are present in the sample, there will be different boundaries which are formed in the buffer. Okay. We cannot see the sample components with the eyes. The boundaries are detected by a detection system. We call this detection system as the Slyron optics. Okay, Slyron optical system. So this system depends upon the refractive index differences in the sample components. Okay, so buffer has its own refractive index, and when the boundary passes through the buffer in front of the camera, then the camera will sense a different refractive index. It will sense a change in the refractive index. Okay, so when it senses a change in the refractive index, it will count it or it will number that boundary as boundary number 1. And depending upon the number of species in the sample, there will be different boundaries. So boundary number 1, boundary number 2, boundary number 3, and so on. Okay, what the camera is doing is it is detecting the velocity of the boundary as it moves in the buffer, right? And based upon the mobility of the boundary, the software in the instrument will help us to find out the characteristics of the sample molecule. For example, what is the molecular size of the sample? Okay, is it clear to everybody?
Yes, sir.
Yes, sir.
So then we move to the next slide. I think I have already described much of the things without making use of the slides, but we'll just try to see what is written on these slides, okay, to get some more information. This technique of moving boundary electrophoresis was invented by the scientist named Tiselius. Okay, and this experiment is also called popularly as the Tiselius U-tube experiment. Okay, so what he did was he used free form of electrophoresis with no supporting medium. Okay, can you see the cursor? Can you see my cursor?
Yes, sir.
Okay,
Yes,
so,
sir.
so I have a habit of moving the cursor along the words. Okay, so as to point to the words, whatever is I'm reading or whatever I'm describing to you. Okay, I'm just trying to point the pointer or cursor so that you know where I am exactly on the slide. Okay, so this sample is dissolved in a buffer and the buffer is placed in a U-tube. I'll just try to show you how the U-tube looks like. Can you see this now? Okay, this is the U-tube. Okay, and within this U-tube, you see these lines here. These lines represent the buffer which is placed in the U-tube. Okay. Then what we do is we apply the electric field. Okay, and when we apply the electric field, electrophoresis will happen. Okay, so when electrophoresis happens, what will happen? all the sample components which are related together, okay, that is the molecules of the same type, they will try to cluster together, they will tend to move within the buffer. Somebody is talking. कोणाच्या माईक मध्ये आवाज येतोय त्यांनी बंद करा. कोणाला काही विचारायचंय का? एनीबडी हॅज अ प्रॉब्लेम? Okay, so the closely related molecules will move as a band or as a boundary. We won't call it as a band, but it will move as a boundary. Okay, and then these different boundaries will move in the buffer, and these boundaries are detected by the Schlieren optics. Okay, what is Schlieren optics? It is a system where we have a light passing through the buffer, okay, and then the light will be refracted as a moving boundary, crosses that a particular region in front of the camera, okay. There will be a change in the refractive index, and the change in the refractive index will be sensed by the camera. Okay, so coming to the applications of this important technique, okay, this is used for testing the homogeneity of the sample.
सांगा ना मला
कोणाचा माईक आहे बंद करा.
काय करू आयाजी?
Okay, so we use this technique for testing the homogeneity of a sample. Okay, what do you mean by this phrase, testing the homogeneity of a sample? Can anybody answer?
So
Same.
Yes.
components are same or not, of same molecular weight or same properties or not, or electric charges.
Homogeneity of a sample. We are talking about the sample. Okay.
Yes.
So what does it mean?
So uniform nature.
Okay, uniformity of the sample in other.
Uniform distribution of molecule in sample.
Yes, very good. Okay, so what we are trying to find out is whether the sample contains a single component or whether there are multiple components of different types in the sample. Okay, so when we say it is a homogeneous sample, it means that only a single type of component is present in the sample. Okay, it is a, for example, you can say a solution of an enzyme, say any enzyme, you can tell me hexokinase. Okay, so there is no other protein present in the sample, so it means that it is a homogeneous sample. And if it is a heterogeneous sample, then it will mean that it contains different types of molecules present in the same sample. Now different types means it could mean anything. For example, it could have different proteins in the sample, it could contain different types of biomolecules in the sample, that is lipids as well as proteins as well as sugars together in the same sample, or it could be anything for that matter. Okay, then what is the next application? Have you understood this application? Moving boundary electrophoresis is used for testing the homogeneity of the sample. What does it mean? How do we know that the sample is homogeneous?
So not several numbers of boundaries will be found.
Yes, very good. Okay, so if we see a single boundary in the buffer in the YouTube, it means that there is a single sample, so there is a single component in the sample. If there are multiple boundaries in the YouTube which are formed, then we know that the sample is a heterogeneous sample, isn't it? Now, why we say that this is an important application of moving boundary electrophoresis? The reason is that we are not seeing this with our eyes. We are using a sophisticated system, a detection system, okay, which is software-based system, isn't it? And therefore, the sensitivity of this particular technique is very high. So even if there are minor differences in the sample components, these sample components will form distinct boundaries, and each of the boundary will be detected by the detection system. And therefore, it is more sensitive compared to the human eyes. What is the next application? Recovery of active biomolecules without denaturation. Can you tell me the meaning of this statement? Recovery of active biomolecules without any denaturation. Of course,
Without
I think yes, go on.
without breaking of bond, sir.
Okay.
Denaturation.
Okay. Yes, you are correct. Okay. So essentially what we are doing is, in this particular technique, we are not going to stain the sample. Like in the gel electrophoresis, I think you have already studied in your TYBSc, isn't it? What is gel electrophoresis? And you might recollect that in gel electrophoresis, after having performed the electrophoresis experiment, after we stop the power pack, then what we do, we remove the gel from the system and then we stain it with some dyes, isn't it? So when we stain the sample with the dyes, then what happens? The staining itself is a kind of a chemical reaction. Your sample molecules are chemically reacting with the dye molecules. Now, if there is a chemical reaction in the biomolecule, or something is happening to the biomolecule, then can we use the same biomolecule in an active form for some application? The answer is no, because we are changing the biomolecule by staining it, isn't it? Then if you remember in SDS page, okay, that is sodium dodecyl sulfate, when we add it, and when we add mercaptoethanol to the sample in polyacrylamide gel electrophoresis, then what we are essentially doing is we are damaging the biomolecules, we are denaturing the biomolecules, isn't it? Of course it has its own advantages, we'll see that later. But what we are doing is, when we are denaturing the biomolecule, we cannot recover the biomolecule in an active form, isn't it? That is the disadvantage of that is the disadvantage of the other gel electrophoresis techniques, but that can be regarded as an advantage of moving boundary electrophoresis. Okay. And then the third application I have written is the analysis of complex mixture. Now, this particular application which I have written actually is a part of the first application we have seen, that is the testing the homogeneity of the sample. These are actually related application. So when I say analysis of complex mixture, what does it mean? Firstly, it means that the sample is heterogeneous. Okay. Already by moving boundary electrophoresis, we can find out whether the sample is homogeneous or not. And then if it is a heterogeneous sample, then we are going to analyze the complex mixture. We are going to analyze the heterogeneous sample using this type of electrophoresis. So what we do when we analyze it? Firstly, we can find out how many boundaries are there. Number of boundaries means we get the number of different sample components in the sample. Okay. And the next thing that we can do is we can find out the characteristics of the sample components. Based upon their movement in the field, we can find out something about the molecular size of the sample molecules, or the charges of on the sample molecules, isn't it? Now, so this is something which is written in red color. Okay, this means that these are the disadvantage of the technique. What is the major disadvantage of the technique? Because it is a free form of electrophoresis. Okay, because there is no supporting medium, or there is no rigid medium which is supporting our samples. This particular technique suffers from the major drawback of convection. What is convection? When the electric current is passing, okay, we can expect that there is some resistance in the sample. Okay, as the electrophoresis proceeds, the resistance goes on increasing. Okay, resistance means what? Resistance to the flow of electricity. So when the resistance increases, what happens is the heating effects are there. The sample buffer, the buffer gets heated up. And when there is heating of the sample of the buffer, then the convection currents are there. Okay, these convection currents in turn result in the distortion of the moving boundaries. The boundaries cannot move along very in a sharp manner. Okay, but there is some disturbance in the movement of the sample, right? And these errors, or these you can say distortions in the movement, may probably result in some errors in the analysis, right? Have you understood this?
Yes, sir.
Yes, sir.
Yes, sir.
Anybody who has not understood this point, okay, you can ask me. You can switch on the mic and ask me. Okay. Then the next important drawback of this method is that it is not a very useful tool as far as the recovery of the sample components is concerned. When see, I've used the term preparative technique. What is the meaning of a preparative technique? Anybody remembers
Mhm.
from your BSc information knowledge that you have already got? This is not a
Uh,
For
preparation.
example,
Genetic
in SGS page,
manipulation, sir.
when
No, no, has nothing to do with genetic manipulation.
sir, for example, in gel electrophoresis, we use fluorescent tagged
Okay.
items or radioactive tagged to
Okay.
recover them and use them for other purposes. So here we cannot do that.
Mm, after having radioactively tagged, how can we reuse it again?
No, sir. Like for in gel electrophoresis, we can tag them and we can recover them, and we can identify them. And here we
Okay.
cannot do this.
Hmm. Okay, so partly
Sir,
you are correct, I'll say, not exactly. Yes, anybody else?
uh, preparation for genetic manipulation, what I had said, means
Okay.
preparing something different kind of genetic changes or whatever
Sir, I
protein
mean for
changes.
amplification using PCR or such things, we
Yes, yes,
cannot
yes.
use
Yeah,
them.
yeah, you're coming closer. No problem. Okay, I can give you a right to your answer. Okay, this particular term, preparative technique, it means that by using this particular technique, we should be able to recover the sample components. Okay, can you see me? Can you hear me? Okay,
Yes, sir.
we are able
Yes,
to
sir.
recover the sample components. Okay, from the maybe it may be a gel or it may not be a gel, whatever it is, and then we can use that particular purified component for a certain application. You're getting me? Okay, so this particular technique is not very useful as a preparatory technique. Why it is not useful? Because if you remember that in this particular technique, the sample components are within the buffer itself. Okay, it is not, we are not separating it from the buffer, and we are not getting it in the form of distinct bands as we get in the gel electrophoresis. Okay, the moment we stop the electric current, what happens is the sample components are going to diffuse again in the buffer. Do you understand what I'm saying? Okay, sample
Yes, sir.
components are in a liquid, they are in a water aqueous based buffer. Okay, as long as your electric current is flowing, the boundaries will be formed, the boundaries will move in the U tube. Okay, the moment you stop the current, the boundaries will also stop. But that is not the only thing that will happen. Once the boundaries stop at a particular place, because there is no further electric current, the sample components are going to get dispersed in the liquid. Okay, they will not remain together as a boundary because it is a liquid. Okay, and therefore if you want to isolate this particular sample components, then it is really not possible, right? So it is not a very, very good preparative technique, you can say. And then another important drawback of this method is that in this particular technique, you can probably analyze, or maybe you can yeah, you can study only one sample at a time. Okay, now I'll show you with the help of the figure what is the meaning of this. Okay, see, this is the YouTube. Okay, in the YouTube we are putting a buffer here, and then probably from this end, using a micro pipette or something, we can add the sample component. Okay, sample is added here, and then we connect this particular YouTube to the electrodes. Can you see the negative electrode and the positive electrode? Okay, so when
Yes,
the
sir.
electrodes are connected and we switch on the current, the sample components are going to move in this tube. How they are going to move? Hmm? The positively charged samples will move to the negative electrode here, and the negatively charged samples in the charge components in the sample will move towards the positive electrode,
Perfect.
isn't it? And then we are going to place the camera. Where are we going to place the camera? Can you see my cursor? Somewhere here, this side, at the center.
No,
Okay,
sir.
now this is left to your imagination.
No,
Okay, yeah,
no,
where?
Yes.
upward side,
Huh? On where?
upward side, upward side,
Upward
Upward
side,
side, sir.
maybe here,
upward side, sir.
here,
Close to that electrode. Yes,
close
sir.
to the
Yes,
electrode,
yes.
but here there is no buffer. Can you see here? The sample is not going to run till this place, is it? Or maybe yes, maybe what you're saying is correct.
The zone formation.
Okay, maybe what you're saying is correct. Okay, now if I keep my sample components here, what is the what is going to be the problem? If I keep the if I keep the camera detection at the center here, then what will happen?
Boundary
Sir,
cache
zone
नहीं करू शकणार.
formation.
See, what happens is, suppose I'm adding the sample
camera
from this
box.
side. Okay. Can you see my cursor? If I
Yes,
am
sir.
adding the
Yes.
sample, just a minute, my internet connection is giving some problem. Can you hear me, everybody?
Yes, sir.
Yes,
Huh?
sir.
Yes,
Okay,
sir.
if
Yes.
I add the sample components here, and just imagine that all the sample components have not mixed up in the buffer properly, because I'm not mixing it. Did I say that we have mixed the sample buffer? I didn't say that. Okay. Even if I shake this tube, I'm really doubtful whether all the sample components will get properly mixed up in the buffer. Okay. Now, when I start the electric current, what will happen? The positively charged sample components will go towards the negative
Negative.
terminal. Okay. Now, if I put my camera here, okay, and then if I say that there are some components which are highly negative, okay, highly negatively charged sample components. So these negatively charged sample components are going to stick somewhere here. They are not going to move to the opposite end. Do you get what I'm saying?
Yes, sir.
Huh?
Yes.
The
Yes, sir.
negatively charged sample components are going to stay near the positive electrode. They will not move towards the negative electrode. Okay. And then if they are not moving to the negative electrode, there is a possibility that they may not cross the path of the camera. The camera may not see them. Isn't it? Okay. So if I place my camera here, then what will happen? Again, the same thing. Again, the same thing. The negatively charged sample components which are unable to move in the tube may not be detected by the camera. Okay, probably I'll have to say something. Even in most of the references, I have not seen anything specific about this particular problem. Okay, but my guess is that probably we have to move the camera along the tube. Okay, the camera should be moved along the tube from one end of the tube to the other end of the tube, so that maximally all the sample components or all the different boundaries which are formed will be detected by the camera. Is it okay? My time is running out. I have about 9 minutes left. Okay. And what I'll do, I'll just ask whether Tikekar sir is going to engage the lectures. Okay. Just a minute. Welcome back. Okay, Tikekar sir will engage the lecture after these about 6 to 7 minutes. And then after his lecture, if time permits, okay, and if you are still not tired, then I will engage the lecture again. Is it okay, everybody?
Okay, sir.
Okay, sir.
Yes,
Yes.
okay.
Yes, sir.
So have you understood what was the problem with the camera? We cannot keep the camera at one particular place because all sample components should pass over the camera. The camera should detect each and every boundary. And therefore, probably it will be more preferable if the camera actually moves along the tube to find out each and every band which is present, or each and every boundary which is present in the tube. Okay. Then we go to the next topic, next form of electrophoresis, that is called as paper electrophoresis. Okay, before we go to gel electrophoresis, we will find out, we'll study what is paper electrophoresis. Now this is probably the earliest form of electrophoresis, and this was used initially for the separation of proteins. Now what actually is done here is a strip of paper is taken, filter paper, and usually preferably it will be better if we use a good quality filter paper, which is you can say a refined kind of a paper, that is Wattman filter paper. I think you have used Wattman filter paper for various experiments earlier. Okay.
Yes, sir.
So what we do is we take a strip of filter paper, and this filter paper actually serves as the support medium. Okay, support medium. On this support medium, the separation of sample components will take place. Now, again, interestingly, I should tell you that this is again a free form of electrophoresis. What is free form of electrophoresis? The sample
Without any
components.
supporting medium.
Yeah, no,
Not
no.
supporting media.
With not content supporting media.
Now, see what I'm saying here. I'm using a filter paper strip as a medium to support, isn't it? Okay. So it looks contradictory. This is contradictory. Okay. So here what we are doing is we are using a strip of paper. On this strip of paper, a thin layer of the buffer will flow. Okay. So filter paper is immersed inside the buffer, and there is a thin layer of buffer over the filter paper. And then when we connect the electrodes and when we start the electrophoresis system, the sample components will move on both sides. Okay, suppose we apply the sample at the center on the filter paper itself, then the sample components will go to both sides depending upon the charges of the components. Isn't it? Negatively charged sample components will go to the positive electrode and positively charged ones will go to the negative electrode. Now this is happening over the paper. But the sample components are not going within the paper. They are within the liquid itself. They are in the buffer. Are you getting what I'm saying? So this is free from a form of electrophoresis because the sample components are not going within the paper. Okay. They are in the liquid, which is over the paper. Okay. And then what could be the advantage here? The advantage is that after the electrophoresis is over, okay, what we do is when we stop the electrophoresis, we put something called as a fixative, okay, or a fixer. Now here we use acetone or methanol as a fixative. What the fixative does is it will try to attach the proteins on the paper. Okay, so whatever separation has happened within the buffer, okay, after stopping the electrophoresis, immediately when we put a fixative such as acetone or methanol, the sample components which are already separated in the liquid will be absorbed by the paper and then they will be fixed on the paper. Okay, so this is something like a combination of a free electrophoresis and a you can say zone electrophoresis, where on the paper you will get the zones of the components which are separated in the electrophoresis. Have you understood what I'm saying?
Yes, sir.
Okay, so I'm coming back to this picture now. Can you see the diagram here? So there are two trays or two reservoirs, and they contain the buffers. One reservoir is connected to the negative electrode, the other reservoir is connected to the positive electrode, and then there is a power pack. So when we start the power pack, there is a flow of current from one reservoir to the other reservoir. And how the current will flow? It will flow through through the paper, which is like a bridge connecting the two reservoirs. Okay, now the actual separation is going to take place over this paper. I'm saying over the paper. When I say over the paper, what does it mean? There is a layer of buffer on the paper. The paper is made wet by the buffer. Okay, and the separation of the components is taking place within the buffer, which is layered on the paper. Okay, now these two things you are seeing here, this part and this part, they are called as the paper wicks. Do you know what is a wick? W-I-C-K, wick. एनिबडी? हा, आपण मराठीमध्ये त्याला वात म्हणतो. वात, दिव्याची वात असते ना. There is a wick we use in a lamp, oil lamp, for example. What does the wick do? What does the wick do?
Ignition with the help of fuel.
No, no, no.
Soak करते सर ते oil में.
Yes, what the wick does is it soaks the oil, okay, and then by capillary action, the oil goes to the end of the wick, and then at the end of the wick is where we actually allow the oil to burn, isn't it? So this is the exact role that this wick is doing. The wick is absorbing the buffer, okay, and the buffer passes over the paper gets wet. Okay, why the buffer is essential? If the buffer is not there, what will happen? Electric current will not pass, isn't it? What does the buffer contain? Do you know? The buffer contains ions. Okay.
Charged
And
particles.
yes, charged ions. And these charged ions are responsible for the flow of electric current. Okay.
Yes.
If the wick is not there, if the paper is dry, then the electric current will not flow. Isn't it? So electric circuit is completed here. Can you see this? The circuit is complete. Okay. Separation
Yes,
is
sir.
taken over the paper within the buffer. Now, these are the different types of images I have tried to put here just for you to understand it. Okay. Whichever image you like, you can take it for you for your studies. Okay. And the third diagram here probably is a schematic representation again, which is a side view. And if there is a question in the exam, if there is a descriptive question, then probably you can draw a diagram like this. Okay. In the diagram, what do you see? There is a paper kept here, paper strip. There are two wicks which are dipping in the electrode, dipping in the buffers. The buffers are kept in the two reservoirs on the left and the right. And then these reservoirs are in turn connected to the electrodes. Is it correct? And the electrodes are connected to the power pack. Can you see the power pack or battery here?
Yes, sir.
Okay. So if there is any difficulty, you can ask me. In 2 minutes, maybe the meeting will get ended. So don't panic. Okay. In about 2 to 3 minutes, Tikekar sir will join us. Okay.
Okay, sir.
Now, the main drawback of this particular technique is that it takes a long time. Okay. Can you see here 16 to 18 hours are required for electrophoresis? Okay. The